Let me start by saying that it's a pleasure and an honor to lecture in this institution, the Royal Institution, where so many of my scientific heroes have stood here at the podium before me. So thank you very much to the committee for inviting me, and thank you very much to all of you for coming to listen. I'm going to tell you about some of the work that we in our lab and others have done trying to understand how the brain maps the world, how it represents a familiar environment, and how it allows us and animals to find our way from one part of an environment to another. So let me start by giving you an example and see how we would do just such a thing. Suppose we found ourselves at Warren Street. What you see in front of you is a map of the area around University College London. And let's say we found ourselves at Warren Street and we were accosted by a visitor. Of course, there are many, many visitors to London. And as we came out of the Warren Street station, they said, could you tell me how to get to University College? And of course, the University College is over here, and Warren Street is here. So we have many ways of doing that. We have many ways of giving them information about how to do it. The most obvious one is to say, I know where I'm going to University College, just follow me. But we're more interested in other ways of doing it. And the two other ways which we think we understand a bit about, and a bit about how the brain represents them, is actually to give the person a route, or to actually use a map and allow them to generate their own way from Warren Street to the University College. So if you give them a route, as shown on the left-hand side here, we might say something like, well, just walk along Euston Road for about 100 meters, and you will come to Gower Street, turn right into Gower Street, and then after about 100 meters, you will be at the entrance to UCL, and just enter, it's on the left, just enter. And that's a series of instructions to walk a certain distance for a certain amount until you see a landmark, and then act in a particular way, turn left, turn right, carry on going forward. Alternatively, if we or they had a map, either the Ordnance Survey map or one of the street guides, we could say, well, here we are on the map, and point to the location of Warren Street. And then we could say, and here's the location of University College London, and you can generate one or many different possible routes to go from one to the other. You could, in fact, generate the same route that I just talked about. You could say, carry on down Euston Road, and then turn right into Gower Street, and so on and so forth. But you could also generate other routes. And that's the important point about maps, is that you could say, okay, go down Euston Road, but if it turns out, as quite often is the case, they're digging up the road for some reason or other, or repaving it or something like that, the map would enable that person to generate an alternative route. They might go up North Gower Street and then find their way around by a circuitous route. And that's one of the most important things about maps, is it is a very, very flexible way of navigating, and it is a way of generating many different routes. And so that if for one reason or another the route that you originally planned is no longer available, then you can take an alternative route. Now, what I'm going to tell you in this lecture is what we know mostly about the way in which maps are constructed in the brain and how they're used by animals. One of the great things, I have to say, working on the part of the brain which is involved in the representation of space and navigation, is that we can draw upon millennia, virtually, certainly centuries of thinking by some of the finest minds in many, many different disciplines. We can look at what geographers have said. We can look at mathematicians' work. We can also look at psychologists and philosophers, and we can get some idea, and it's a great, great repository of information and a wealth of knowledge. Well, I thought I'd just start by giving you a few instances of what psychologists and philosophers have thought about space and how we represent space. For many years, starting around 1900, and particularly in the United States, there was a large industry of what were called, to some extent called slightly derogatorily, rat runners. People who were trying to understand, psychologists mainly, were trying to understand how rats found their way from one part of a maze to another. And what you see on the top of the screen there is a typical maze, and I'll come back to that in a second in my lecture. Many of the psychologists, particularly those on the east coast of the United States, looking at how animals behaved in mazes, concluded that the way they did it was primarily by following routes, exactly the sort that I showed you. Go down to the first choice, turn left, go to the next choice, turn right, and so on and so forth. But others, primarily on the West Coast, and particularly this man, E.C. Toulmin, who was at Berkeley for most of his career, believed that rats had some sort of a more sophisticated cognitive representation of the environment, and they could use that to find their way around. So one of his famous quotes is shown here, in which he said, "We believe that in the course of learning, something like a field map of the environment gets established in the rat's brain. Incoming impulses are usually worked over into a tentative, cognitive-like map of the environment. And it is this map, this tentative map, indicating routes and paths and environmental relationships, which finally determines what response, if any, the animal will finally release." So he's saying, yes, we don't deny that they could learn left, right turns, and so on and so forth. But we think they also have some sort of a cognitive representation of the environment, and they can use that to read off the particular route. Well, as you might expect, over centuries, the philosophers have also had something to say about the way in which we represent space. And about 4 or 5 years ago, I gave a talk here in a smaller room on the philosopher Kant. Because Kant, following on from Plato, actually thought that when we are born, we come into the world with certain ideas about the world and how we should represent it. We come into the world with a framework which actually allows us to organize the sensory inputs as they come in. And actually, he thought that one of the major organizing principles was the notion of a three-dimensional Euclidean space. So he said in many, many places, and I've just summarized it, that three-dimensional Euclidean space is a form imposed on experience by the mind. You don't learn about space, you don't learn about three-dimensional Euclidean space from interacting with the world. You actually come into the world in some way, and of course, this is now we're talking about the 18th century. He didn't actually know how it could happen, but being a Platonist, he thought that you could actually come into the world with this representational, this powerful system, and that you would use that to organize the incoming stimuli and also to organize your behavior. He had an arcane language, and I'll quote some of it, but what he said was, space is nothing but the form of all appearances of outer sense. So if you want something to be, and understand that there's something outside there in the external world, then it has to be represented in space. It is the subjective condition of sensibility, the receptivity of the subject, the capacity to be affected by objects. And it must necessarily precede all of intuitions or all understanding or sensing of objects. And further down he says, as it exists in the mind a priori, it can contain, prior to all experience, principles which determine the relationships of these objects. So he believed that your ability to represent the space of this room, the space of London, your ability to navigate around, in some ways was there before any experience, and it was used to actually organize your experience. Of course, the opposing idea was if anything, more popular, both with the British empiricists, and I've chosen a quote from Whangarei, the great French scientist philosopher. From his point of view, this idea that there's space in the brain and that you can represent an absolute is just nonsense. And it's necessary to begin by referring space to a series of axes invariably bound to our body. So what he was saying is that you don't have this representation, you don't have this abstract representation, you just can construct a kind of type of space on the basis of your experience where things are relative to your own body. Directions of movement were not given, according to him, and here I think I will be able to show that he was wrong. But the sensations which correspond to movements in the same direction. So if I'm going to move over there, I might go this way, I might go that way. And the brain somehow or other puts these all together and adds them together. And that is what you mean by the sense of direction. So to localize an object in space simply means we represent to ourselves the movements that we must take to reach that object. So we have two of the great giants, the great thinkers of philosophy coming down on very different sides of, do you use maps or map-like representations, you have absolute sense of space, or are you really just reduced to route-like strategies to knowing that you move to a certain location and then you make a left or a right turn? And I think, as is always the case, they're both right. And I think the person, the philosopher who captured it best is a British philosopher, Stephen Toulmin, who spent most of his time in the United States, in Chicago, and who was really interested in the philosophy of science. And as part of his writings in this book, The Philosophy of Science, it's a very small primer, but it has more insights into many of the dilemmas of philosophy than many other great big tomes. And what he said was, look, folks, it's pretty obvious you have both maps and you have routes. And what he set out was what really are the differences between the maps and routes and how they interact with each other. And he said, for example, to a man making a map, all routes are as good as each other. So if you want a route, you really want to know where you're going and what the endpoint is. But if you have a map, you can generate, as I already showed you, an infinite number of routes from it. The users of a map will not be all going the same way. So a satisfactory map is route neutral. So there must be a representation which represents the world in a way which doesn't presuppose how you're going to use the map and exactly how you're going to get from one place to another, or even which places you want to go between. An itinerary, or a route, however, is specifically concerned with particular routes, starting points, and destinations. And the form it takes is correspondingly unlike that of a map. Often enough, of course, a map may be used to work out the itinerary for a particular journey, and from one map, an indefinite number of routes may be read off as occasion requires. So what he's saying is that you probably have both, and there must be some way of going from one to the other. And as I showed you in the first slide, if you have an adequate map and you know where you are and you know where you want to go, you should be able to read off a very large number of routes from those two. Okay, so our group has been interested in both the way in which the rodent brain creates and represents and uses maps, and also how humans would do it. And for a long time, we struggled with trying to figure out how do you test the use of map-like representations in human beings, and how do you assess which parts of the brain are involved in it? And it was only until the middle of the 1990s that my colleague Neil Burgess and I, and our psychologist colleague, Eleanor McGuire, realized that the way to do it was to use virtual reality. Now, we knew that virtual reality was probably a good way of doing this, but up until that point, virtual reality systems were actually only used by automobile manufacturers, Hollywood filmmakers, and so on and so forth, and they were extremely expensive. in the 1990s, what happened is we got these first-person shoot-'em-up games, which all of the kids, certainly in the 90s and in the turn of the century, were using to go around and get guns and get weapons and kill monsters and do all kinds of heinous things. So what we decided that we didn't really want all that other stuff. And what we would like to have is to have one of these games where we could extract just the spatial aspects of it and see if we could use that to understand how the brain represents space. So Neil Burgess, to his credit, spent several months taking apart one of these games and getting rid of all the unpleasant aspects, or getting rid of the monsters and getting rid of the guns, and so on and so forth, and left us with what you see in the middle of the screen there, the sketch map of an environment which had all sorts of aspects to it. It was about 70 meters by 70 meters, and where people could wander around in this environment, in the virtual world, and learn about where they could go to a film, as you see on the left-hand side, or where they could go into the theater and what film they would see there, or where they could get a drink, or where they could go and play billiards. And it takes about an hour to find their way around in this environment. And then what we did is, of course, we put them in a scanner. In those days, we were looking at blood flow, and now we look at fMRI. And so Eleanor then ran an experiment in which she showed people who knew this environment where they were in the environment, they could see where they were, and then where they should go. And there were several conditions, and I won't go into the details of it, but one of the conditions, they had to actually access their representation of the environment and try to go by the best route. And what she found is shown on this slide, which is that when we looked at the brain and which parts of the brain were active when people were using this map-like representation as opposed to either following a route that was laid out on the ground or some other device, there were very, very few parts of the brain which were involved. And one of them is the one shown here. It's called the hippocampus. It's shown on the two sections on the right-hand side. And you can see little white areas. What's great about all this imaging stuff is you end up with little blobs on different parts of the brain, and you assume that means that part of the brain is more active, even though you're looking at an indirect measure. And the part of the brain that's shown here is called the hippocampus, and I've used several schemas, shown several schemas on the right-hand side, and where it's shown in red. And if you look at the rotating brain, you'll see that it's sitting inside the temporal lobes here, and it's a long, extended structure. And if you look at it from the right angle, in the right direction, on a good night, it looks like a seahorse. And that's what Orontius, when he first discovered it and dissected it out of the human brain in the 16th century, he said, looks like a seahorse to me. And so he called it hippos campos, which is the Greek for seahorse. There was one other part of the brain, which is not shown on here, and we know now that part of the brain has something to do with the generation of roots. And these two parts of the brain talk to each other. More importantly, we could look and see if somebody who was trying to find their way around in this environment was doing it very efficiently, going directly from one place to another, for example, as shown here in the yellow trace, or was doing it inefficiently, as shown in the green trace, or was getting completely lost. And it turned out that this part of the brain, the hippocampus, became more active when the person was actually using the best strategy and going directly from one place to another. So there was not only evidence that it was being used, but it seemed to increase the amount of use that it was being subjected to in terms of the activity, as shown on the plot on the right-hand side, which shows the amount of activity on the x-axis and the accuracy of the actual navigation on the right. Eleanor then realized that she had a gold mine. And she is the one, and it's fully to her credit, who realized that maybe if you could find a set of expert navigators, their hippocampus might be different from those of the rest of us. And so she realized that in London, the taxicab drivers are expert navigators. They have to pass a test, and this, of course, is now becoming more of a historical lecture, because as we get GPSs, you will notice now that many of the cab drivers are surreptitiously looking down at their iPhones as they're navigating. But until a very recent time, to become a cab driver, you actually had to pass a test, and it meant you had to have a knowledge of 25,000 streets in London and how to get from one to the other by the best route, taking into account time of day, traffic conditions, and so on and so forth, weather conditions. So Eleanor did what now seems, in retrospect, obvious, but at the time was really quite an important insight. She said, well, let's go and look at their brains. Let's forget about the activity in their brains. Let's just look at the structure of their brains and see if they have certain parts of their brain which are different from other parts and which are different from non-taxicab driver subjects. And what she found was, as shown in this slide, is that the one part of the hippocampus is larger in taxicab drivers than it is in non-taxicab drivers, including bus drivers. So including people who have tried to go back and forth around the streets of London, but do so in a very stereotyped, route-like way. It was different. There's no free lunch, and there's no free lunch in the brain. So the fact that they had a larger posterior part of the hippocampus was coupled with the fact that they have a smaller anterior part. And it also turns out that you're not born to be a cab driver. You don't come into the world with a big hippocampus and saying, I'm going to be a cab driver. And that's shown very clearly on this slide, where if you correlate the amount of time that somebody has been driving a cab with the size of the hippocampus, is a good correlation. And I'm not showing it here, but it turns out that when people stop being cab drivers, the hippocampus slowly shrinks back over time. So it certainly is a question of use it or lose it. And we now know that many parts of the brain are like the hippocampus. They're like muscles. The more you use them, they grow. We don't know exactly what the neural basis of this is. We don't know whether it certainly is unlikely to be more cells being added. It's more likely to be different parts of the cells are being added. Okay, well, that's how you study parts of the brain, and particularly the hippocampal areas in humans. For many years, we've been studying also this part of the brain in animals. Firstly, looking at their behavior, if there's damage to this part of the brain, but also at the same time looking at the cells. And that's what I want to tell you a bit about now in the next part of the talk. So very soon after we started thinking that this part of the brain might have something to do with spatial navigation, Richard Morris, who's now at Edinburgh, came into the lab and said, okay, if you really think that a rat or a mouse needs this part of the brain in order to find its way around the world by novel routes or by different kinds of routes, starting from different locations, going to the same location, then I think I've got a test for you. And the test he came up with is shown on this slide. What it is, as you can see on the lower left-hand side, it's really a big swimming pool. It's a swimming tank of about 1 meter in diameter. And the swimming pool is filled with slightly opaque water, milky water, and somewhere in that pool is a little platform which can be either above the level of the water or below the level of the water. If it's above the level of the water, then the animal can say, oh, there's the platform, and he can swim straight to it. If it's just dropped a little bit down, and so it's below the level of the water, he can't see it. He then has to localize himself, say where he is in the room, find out where he is, and then remember where the platform is in the same coordinate system and swim to it. And I wouldn't be telling you this if it didn't turn out that animals with damage in this part of the brain, as shown on the lower right-hand side, where you see several traces, had great difficulties doing this, what appears on the face of it to be a very simple task. So the middle trace shows you what an animal with damage to its hippocampus does, and the two right-hand traces show you what control animals do. But let me give you some idea of the actual, of what it looks like. Because Richard has put a very nice video on the web. There's the pool, and you can see the hidden platform, which is somewhere to the left of center. And there's an overhead tracking system. The animal has just been placed down, and the animal swims. This is now an animal who knows his stuff. He's been at it for a while. Of course, we want to make sure that he's not using anything coming from the platform, so that he's using the information. So you essentially get rid of the platform. You start him from a new location, and he goes straight directly to the location where the platform should be. And of course, he can see he swims around looking, where's the platform? Yeah. Rats are very clever, as are mice, and eventually he says, I know, this is a psychological experiment. They've gotten rid of the platform. So they moved to someplace, and you can see he starts to sort of wander off looking around for where the platform has been moved. And that's one of the things we know is that at least in terms of spatial abilities, rats are very good. Okay, now that test is probably the most widely used test in all of behavioral psychology. You cannot go into a drug company without finding several water mazes. You cannot find any, there are tens of thousands of citations in the literature to people using it for all sorts of purposes. And anything that anybody wants to use, where they want to use a behavioral test to assay damage to the hippocampus, as I'll show you later on, we're particularly interested in looking at subtle damage to the hippocampus, they will use that test. Now, I've already alluded to the fact that there's a whole history of maze learning, starting around 1900 with Small's maze here in the United States. And we can use a lot of that expertise to look at the water maze and ask, is this as good as we can do? And what my lab has been doing recently has been to try to take the basic idea of the water maze and improve it. See, can we make it better? And can we make it more maze-like? It's called the water maze, but it doesn't quite fit all the canons of what a maze should look like. The original maze was devised by Small, and it's shown on the upper left-hand side here, and is based on the Hampton Court maze. So his research director said, go out and make a maze. So he went and he looked, and the first maze he found was the Hampton Court maze, which is shown on the lower right. And you can see people kind of wandering around in it still today. And he kind of linearized it. He made it a little bit easier to construct it by, not of hedges, but of course by wooden blocks. And you can see that in his version of the maze, the animal can wander around, and there are many different ways it can go, and many different cul-de-sacs and things like that. And over the years, particularly in the United States, there was a development of maze technology until in 1925, the maze you see here, Stone's multiple T maze, was constructed. And the statisticians started to play a role. And what they say is, look, what you want for a really good maze is to be able to put the animal into the maze, and that every time it makes a choice, that doesn't in any way hamper its ability to make a successful choice on the next trial or on the next choice. So if the animal goes wrong on the first trial, the first choice, then you want it to be able to have a good chance of going correct on the next trial. And you would like it to be a series of binary choices. So thinking about those sorts of things, over the last couple of years, my colleagues and I, some of whom are here in the audience, have tried to take the water maze and turn it into a proper maze, into a maze which consists of a series of binary choices, to see if we can understand how rats solve the problem, how normal rats solve it, and how they're impaired if they have damage in this part of the brain. So what you see here are several pictures of the maze. It consists of 37 platforms. Each one is individually raised or lowered. And you can see on the upper left-hand side, all of the platforms are down. On the upper right-hand side, one of the platforms is raised. And when we put the animal on that, as you'll see in the video in a second, it's confined to that situation and can't go anyplace. If we then raise 2 more platforms, it can choose between them. So it has a binary choice. So instead of giving the animal a complete freedom, as the water maze does, to go in any direction that it wants to, we force it to take a particular choice. And so we say, you're now here at this location. What do you think is the best place to go next if you want to get to the goal? And so as shown on the graphics here, we can, for example, on the left one, we can give the animal who's sitting on the blue platform a choice between the yellow or the brown maze platform. And so you can see that if the goal is where the red platform is, then if the animal understands the structure of the task and understands the structure of the maze, it's a no-brainer that it actually should choose the yellow one, because that's taking it closer to the goal and it's taking it in the right direction. And we can make it more easy, or we can make it more difficult by changing the relationship between those choice platforms. So for example, on the right-hand side, you can see that if the animal's on the blue platform and we give it a choice between the yellow and the brown platform, neither of which is really a good choice, it still should be able to say, well, if I got to choose one of them, I better choose the yellow platform because that's taking me less far away. And it turns out, as I'll show you in a second, normal rats can do that. And we think that's telling us that they have a pretty sophisticated geometrical system for calculating directions and angles. And I'll give you some evidence from the cellular work that shows that. Okay, well, let's take a look at what a representative trial looks like. Here's the animal. He's sitting there. He knows where the platform, the goal is, which is down in the lower right-hand side. And he sits there and says, okay, I know where I want to go. And then he looks around and he says, oy, oy, oy. You're giving me a choice between something going in the opposite direction or something which is only 135 degrees away. And so he chooses that one. He chooses something which is better. And then, of course, we can force him to go where we want. We can say, okay, we only give him one platform, he goes there. And as you'll see now, it gets easier. We're more lenient and give him better choices. And he now chooses the better platform. Remember, he's trying to get to a platform on the lower end side, and he waits for it to come up. He looks around. But when the platform comes up, he knows exactly where he is. And sometimes he hesitates. And many of my colleagues are interested in decision making. And you can see here, you can almost see him thinking. You can almost see him thinking, is this the right one? Okay, this is the right one. And he looks around, and he's not running he's not running rapidly, he's taking his time. When he gets to the goal, there's no food there. Psychologists have all sorts of tricks, because we don't want him to have the ability to smell or have any other information coming from the food. So it's only when he gets there that we put the food on the platform after a few seconds. We're not that bad. Okay. So I won't go into the details of it, but just to summarize this slide, saying that normal animals learn this task very quickly. They learn it as quickly as the water maze in a very small number of trials, as shown on the upper right. Animals with damage in the hippocampus really find it very, very difficult, and they never, ever get to more than about 75%. It turns out that normal animals also are sensitive to various aspects of the task. So they're sensitive, as shown on the lower left, to how far away the choice that they're making is from the goal. The farther away they are, the harder it is. And animals with damage to this part of the brain are actually even worse at making that discrimination. And finally, the normal animals are also sensitive to how large an angle we give them between the best choice and the heading direction to the goal. So if we give them a choice between a platform which is heading straight to the goal and another one, they're very good. Whereas if we start giving them choices of the type that you saw on the first choice there, where they have to choose between two difficult ones, they're much less good. But they still are better than chance. They still know how to choose between two very difficult ones, which are both taking them away, but one was better than the other. Okay. Now, so it's clear that both humans and animals can use something that you might think was like a map. And what we would like to know then is, what does the map look like? And I have to confess, we started off knowing a little bit about what the map looked like before we went into this whole enterprise. We knew from work that Jonathan Dostrowski and I had done many years ago that in the hippocampus there are cells which represent the animal's location. And I've shown you a modern day way in which we study these cells and how we actually try to work out what they do. So what you see on the upper left-hand side is what the rat hippocampus looks like. You've already seen the human hippocampus. And we know from anatomy and physiology that in many ways, these two structures are very, very similar. And people sometimes say, why would you use an animal to study something about the human brain? Or what can knowing something about a part of the animal brain tell you about the human? But of course, one of the great things about studying space and navigation is these are things which we have in common. And it may be difficult for us to understand very much about human language by studying a rat, but it certainly is true that if we can understand how rats navigate, how they represent the spatial world, then we think we have made quite a bit, as I'll show you in a moment, of progress about how humans do it as well. What we do is we place very fine wires into the parts of the cellular parts as shown in the middle. And this enables us to actually record from the cells as the animal goes about its business. So the animal is operated under anesthesia. We give it many analgesics so that it's comfortable after the operation. And then after about a week, we can look at what the cells are doing, what they're interested in as the animal goes around the environment. And we typically do this as shown in the upper right-hand corner, where we let the animals explore and look for food in an open box. We try lots of other things. We give it little tasks and so on and so forth. But for the purposes of this lecture, the most important thing about these cells is that they respond when the animal goes to a particular location. And let me show you another slide here. This is a video that we've made. It's a cartoon that's been made. It's real data, but it shows this little cartoon rat running around in this, now in this case, a circular environment. And what you'll see is little red dots being dropped every time one of these cells fires. So we're looking at just one cell, and we're interested in what this cell is represented. And you'll see that the cell is particularly interested when the animal goes over to a particular location. It doesn't care why he's going there. It doesn't care about many other things, what he's doing there. In this case, he's doing the same thing all over the environment. He's actually looking for food and eating it when he finds it. And the food is thrown in randomly, so that gets him to study the whole maze and to explore the whole maze. So here's a cartoon, which was made by one of my colleagues, Yulia Krupic, and you can see that in general, the cell is very quiet. And if you didn't have the right task, you would never know that the cell was even there. But every time the animal goes over to that central left-hand area, the cell fires. And it doesn't make any difference whether he's heading north or south or east or west as he goes through that area. The cell only cares about some area of space. It's not responding to any simple sensory cue. We know that these cells use lots of different sensory information, but they're using them to compute a location in space. So the first story, and this is the first thing that we know, is that the idea of space is not reducible to any simple sensory modality. The idea of space is something that's constructed by the brain very much in the way that Kant thought it was. Okay, now that was one cell. If we look at a whole bunch of cells, and one of the things that's very exciting about the field of neuroscience, and particularly systems neuroscience these days, is that we are getting many techniques for looking not at a single cell and what it's doing, but for whole groups of cells. So here's a situation in which we're looking at 30 some odd cells. We can now look at hundreds and hundreds of cells. I don't have time to go into the various techniques. We can talk about it in the discussion. And what we see is just looking at 32 cells, is that each one has a different favorite part of the environment. So you see that the one that I've represented on the upper left-hand side is interested in the upper left-hand side of the box. And the one on the lower right-hand is interested in the lower right-hand side. It turns out that's a bit of a fiction because cells that are right next to each other might be representing very different parts of the box, and cells that are very far away might be representing very local things. So there's no simple representation of an environment in any sort of simple map-like way where you would say, I know that I'm over here, and then that means that the cell next to that is going to represent the area next to that. And we're still struggling a bit trying to understand why that is the way it is, but it is in fact the way it is. Okay, so we've known for a long time that cells in the hippocampus itself, in the hippocampus proper, represent locations. And different cells represent different locations. So you might think, okay, that's getting on for looking like something like a map. You have every place in an environment, in a known environment, has its own cell. In fact, it has lots of cells which represent that location. But if you think about it a bit, having a lot of representations of places doesn't give you a map. Because what you need in addition to have a map is shown in this cartoon here. What you need in addition is to know the spatial relationship between those locations. It's no good knowing I'm here, but then not knowing what happens if I go over here. The system needs to know that there is a spatial ordering, a spatial relationship between the different places in the map. And that, we think, is given primarily by a system which records the movements that the person or the animal makes and relates things according to those movements. It essentially takes the movements, such as turning by a certain number of degrees or walking a certain number of human steps or rat steps, and putting them together and creating what I'm showing here on the upper right-hand side as a series of vectors. So we think that something like the map looks like a group of locations, representations of locations of place cells, A, another group B, another group C, and the relationship between them is represented by vectors which represent the distance and direction between those two. And having something like that will fulfill most of the requirements of having a map. Because as you can see, if you wanted to go from A to B, you would choose the vector AB. But if that were blocked, the system should also generate the vector AC and then CB, and by a little bit of vector algebra, you should be able to get back to the same place. So the idea is that the system is always calculating all the possible routes to go from where you are to where you want to be. Okay. Over the years, in our group and in many other groups now in the world, we've been looking for those types of representations. We knew that there were these place representations, but we needed also, if we were going to be able to at least make some progress in how the map worked, we need to know that there are these other representations of distances and directions and things like that. And I'll just summarize and show you a couple of slides to give you some feeling for what some of these other spatial cells look like. What I've shown here is a cross-section through the rodent hippocampus, and shown here is the hippocampus proper, where we find the place cells. But we now know that there are a whole series of other areas which are tightly interconnected anatomically to the hippocampus, and primarily areas called the subiculum, parasubiculum, presubiculum, and this area here, the entorhinal cortex, and they also contain spatial cells, and they all talk to each other. I won't go into any details of the anatomy, but they're all talking to each other, and they're certainly all sending information into the hippocampus to the place cell representation. So if we just go around the outside of this, the place cells are found, as I say, in the hippocampus. If we look at the next set of cells, we know that there are cells which represent the animal's distance from landmarks. How far is the animal for, say, a landmark being, say, the edge of the box? How far away is it from the east wall of the box? And some cells like it to be very close, and some cells like to be very far. If you then carry around the circle, you see that there are some cells called grid cells. And they are cells, which I'll come back to, which look like they, at least on the first glance, are providing information about the distance in particular directions that the animal has moved. And you can see they look like a whole series of tiny little place fields. And I'll show you a little bit more about those in a second, because we originally got very excited when we saw that. Because coupled with the final set of cells, the head direction cells shown on the lower left-hand side there, which seem to be telling the animal the direction in which it's pointing, we thought, and this is around about 2005, we had all of the bits and bobs, all of the components of the map. But as I'll show you, we're not out of a job yet. There's still a lot more to learn, and we still have to do more work to try to understand particularly what the grid cells are doing. Okay. I'm going to skip this part about the head direction cells. I think I'm running a little bit late. These are cells which don't care about where the animal actually is in the environment, but they care where the animal is facing. So you record from one of these cells, and the animal faces to the northeast. They're not geomagnetic, but if he faces in a particular direction, then the cell will fire, and it won't fire if he faces in any other direction. And he doesn't care where he is in the environment. Doesn't care whether he's over there or over here or there. So they're doing a sophisticated computation, and they're creating the abstract notion of direction. And we think these are the ones which are telling the system which direction the animal is in. And importantly, if you look at a bunch of these cells as shown in the lower right-hand side, if you have two of them, and you confuse the animal, let's say you have one that's pointing north and one that's pointing east, and you do something which confuses the animal momentarily, and that the one that was pointing north starts to point to the west, then what you find is that they've all rotated by the same amount to maintain the internal consistency, the cohesion within it. So they're acting like a compass. And they're acting to tell the animal that within some framework, the framework of the room, the animal is pointing in a particular direction. We also know that, as I said, there are cells which respond to boundaries and distances from boundaries. And we had predicted this as a result of some of the earlier work that we had done. And Colin Lever in our lab went and looked for them in a place we hadn't looked before, in the subiculum, one of those other areas. And these are cells which look very much like they had been in the minds of the abstract expressionists, like Barnett Newman or Mark Rothko, because what they do is they delineate an area of the environment, as shown, for example, in the Rothko on the upper right-hand side, and says that this is now a certain distance from the edge in the painting example of the canvas, or in the brain example, a distance from the edge of the environment. Okay, now I've told you that we think we have cells which actually are delineating something about distance. And the question has always been, as you can see on the right-hand, upper right-hand side, whether distance in the brain is measured in some way, such as Blake's depiction of Newton, where he has a calipers and he's actually trying to measure off the distance between 2 points on it, or whether it's something more sophisticated, as shown in the Roy Lichtenstein paintings. Roy Lichtenstein, his signature, his signature way of painting things is to paint a nice painting, and then to lay on top of it this series of Ben-Day dots. And as you can see here on the right-hand side, he's taken what the painting called the yellow cliffs, and he's actually laid these dots on it. And if you look at these dots, they essentially are a Cartesian coordinate system laid on the structure of it. And you can imagine that if the brain had something like that, it would actually be able to compute how far an animal or a person had gone in a particular direction by just counting the number of dots. So you could see, for example, if you wanted to see how far you were going up the cliff here, you could say, I'm here and I want to count several dots up, or you could count in an orthogonal direction. It turns out, as we now know, that the brain uses the second type of representation. We don't know that it doesn't use the first type of representation, but it certainly has something like the Liechtenstein pattern laid out across the surface of every environment that the animal finds itself in. And we know that because of the work done in the lab of the Mosers, as shown in the picture, Edvard and May-Britt Moser. And what they found is these cells, which they call grid cells, and what you're seeing is the type of pattern that is laid out by one of these cells as an animal moves around the environment on the upper left-hand side here. These cells have not just one preferred location in the environment, but multiple preferred locations. So as the animal goes around, and you can imagine, here's an animal, I won't show you any videos of this. As an animal runs along, for example, from the lower left-hand side here, up in that direction, the cell will go brup, silent, brup, silent, brup, and so on and so forth. So you can imagine that the cell, other cells in the brain could count the number of brups, 3 rat feet or something like that, and that this would be the metric for the map. And one of the things that they showed is shown on the right-hand side of the thing was that different cells, particularly at different parts of this area called the entorhinal cortex, have different spacings between the cells. And it looks as though the spacings are not uniformly distributed, but they're quantized. So as you go from the upper part of this area, the entorhinal cortex, you get very tiny spacings between the peaks of the grid firing. And as you go deeper, the spacing increases. So it looks as though the brain has constructed a way of measuring distances in the environment and actually using different methods, which presumably one could use to measure the distances in different size metrics. Okay, that's where we were. We thought we have now all of the spatial cells we need. Bring in the mathematicians, bring in the computational neuroscientists, they can construct mathematical models and Bob's your uncle. A bit too early. We put the champagne back in the bottles because Julia Krupic and Marius Bauza noted that all the environments that we and the Mosers and everyone else who had studied these cells were all highly symmetrical environments. They were all squares and circles. And whereas we would like to have believed that these cells didn't care too much about the environment, they would measure out the same distance in any environment, it turns out that Marius and Julia were able to show that the shape of the environment is an incredibly important aspect. So we thought, look, these cells are probably there right from the beginning, and they're actually doing this computation in any environment. Turns out, not the case. And so what they did was they recorded from animals in squares, as you can see on the right-hand side, and in non- or quasi-symmetrical environments, such as trapezoids, as shown on the left-hand side. And what they found was that whereas in a square environment, you get this beautiful symmetrical pattern, which is the same on each side of the box, as shown on the upper right-hand side, when you put the animal into a less symmetrical environment, the trapezoid is still symmetrical, but only around one axis as opposed to the square, then what you find is that to one end of the trapezoid, the pattern still looks very much like what you see in the square, but as you go towards the cone of the trapezoid, the pattern starts to break down. It's almost as though the pattern is being squeezed and elongated by the structure of the environment. And so if you look at, as shown in the graph, if you look at the symmetry between the two sides of a square, the pattern in the two sides of the square, it's very similar, whereas the symmetry is broken down in the trapezoid, and the two sides are actually measuring different things. And the pattern on the right-hand side on the left-hand side of the trapezoid is really nowhere as symmetrical and regular as it is on the right-hand side. They and others have gone on to look at this much more systematically, and instead of just looking at 2 boxes, they've looked at 4 boxes where they systematically change one aspect of the environment to go from square rectangles to polygons and so on and so forth. And I won't go into it in any detail, but it looks as though, as you can see on the right-hand side, if you just look at the arrows, here, this cell, this field here, as this wall changes, this field is being pushed up in front of the wall, whereas when you look at what's happening on this side, where there's no change in the structure of the environment, nothing's happening. To summarize, the pattern, this beautiful symmetrical pattern, only occurs in beautiful symmetrical environments. And of course, as we all know, if you look around here, this is not a beautiful or not necessarily perfectly symmetrical environment, and many of the environments which animals and ourselves find ourselves are not. Okay, I'm almost out of time. I have about 5 minutes. I want to just finish with a question which has been in the back of our mind for a long time, and which we're now beginning to think about and actually do some work on. What does this all mean? I mean, is this going to be of any relevance? Is it going to have any clinical benefit? When we first started working in this area, I have to say, we did so because I was interested in, and my colleagues were interested in, trying to understand how this part of the brain works. It's pure curiosity-driven research. But over the years since then, it's emerged that this part of the brain, the hippocampal formation, is one of the first areas attacked by the toxic processes that take place in Alzheimer's disease. Alzheimer's disease, as you know, is an epidemic, and you can see from the graphs there that as you get older, you will find that there's an increasing susceptibility to Alzheimer's. It's true in all parts of the world, and the figures across the world are about something like, as it says here, 4% of everyone who's over 60. And in the field, there's the belief that if you live long enough, you're going to get it. And so the reason most people don't get it is because they die before it reaches a certain age. It consists of the movement, as I say, of toxic proteins which look like they first manifest themselves in this part of the brain, in the hippocampal formation. And people like the Brocs have actually staged taken the different stages of Alzheimer's and looked at the brains of patients who happen to have died for other reasons at the different stages. And what you see is shown on the very far left is that the disease manifests itself as amyloid plaques, and below that as neurofibrillary tangles, which consist mostly of tau. And if you look at the pattern of tau and the expression of tau, you see that at the very early stages, when people are just beginning to show memory problems, just beginning to lose their way, the tau is confined almost entirely to the hippocampal formation as shown in the orange on the first set of brains. And then as the disease spreads, and we don't quite know how it spreads, what you see is that the tau manifests itself in larger and larger areas. And we actually think it's moving along synaptic connections. So it moves first into areas which are connected to the hippocampal and the hippocampal formation, and then from there out into the cortex. And at each stage, you can correlate the actual manifestation, clinical manifestation of the disease, the memory problems and the getting lost and things like that, with where the disease is in the brain. So thinking about that, we thought, well, if we really believe that we understand what these areas are doing in humans and rats, we should be able to do two things. First thing we should be able to do is we should be able to come up with very sensitive tests for the earliest stages of Alzheimer's. And we know that there are patients who have mild cognitive impairment, about half of whom will go on to develop the full-blown disease. We know we have biomarkers which begin to show up very early on. We also know that one of the first anatomical signs is that the hippocampus starts to shrink a year or so before people show clinical signs. But that's not good enough. We really need to be able to identify patients at risk, people at risk, 10 years early, because any of the therapies that, if we ever going to get them, we have to be able to start using them well before we get the clinical manifestations. So colleagues at Queen Square have been trying to come up with very sensitive spatial tests. And I'll just show you one or two of them, and then I'll show you a little bit about what we know from the animal work. The first is a very simple test. It's shown on the top part of the picture here. You show someone a picture of 4 mountains, and they have a different size, and they have a topographical relationship to each other. and they're seen from a particular perspective. So they're seen from a particular point in space. And then what you say to them is, okay, I'm going to show you 4 other pictures, and you have to tell me which are the same 4 mountains as seen from a different perspective. So make believe you walked around the mountains and you're looking at them from a different perspective. And that's the 4 pictures are shown on the right-hand side. And it's not an easy test to do. You really do have to do something like create a representation and then be able to rotate it or imagine yourself walking around. We know that it's very sensitive to hippocampal damage because patients, as shown in the lower graphs on the right-hand side, patients with frank hippocampal damage, either because they've had surgical removal or because they've had developmental problems, are very, very bad at it, particularly when it's a memory test. So you show them the picture on the left first and then wait 2 or 3 seconds and then show them the one on the right. But even in some cases, when all the pictures are right there in front of them. So we're trying to develop tests like that. We're using virtual reality to see if we can identify patients who are at risk of Alzheimer's and see if we can then predict which ones are going to go onto it. And that test itself is actually quite good in terms of its correlation with biomarkers. Patients who have biomarkers which we know predict Alzheimer's actually, as shown on the middle, on the second one from the left, are actually much worse, it's the third one from the left, are much worse and much more like full-blown Alzheimer's than other patients at risk. And finally, I just want to tell you that given that we understand a lot about the physiology of these cells, and given that we understand some of the genetics of the disease and what predisposes people to get the disease, we should be able to generate animal models, at least partial models of the disease. And we've done that, and other people have done it, and we've been testing those animals for some time now. And one of the things we want to see is what happens to the place cells. If we take a group of animals which are, we think, at least partial models for the disease, and we let them get older, because aging is, of course, one of the major factors, what happens to the cells? And the results of a study we did some years ago, in which we looked at animals which had changes in their expression of amyloid. You can see on the left-hand side that when those animals are young, the fields look very much like normal fields. They're nicely formed, and they have discrete areas of the environment that they actually code for. As the animals get older, then the wild-type animals, the normal animals, still show very nice, orderly place cells. But some of the animals who have this mutation are beginning to show cells, as shown on the far right-hand side, where they look like they're just much less good at identifying the animal's location. They don't look like they're doing their business so well. And they sometimes fire in several places, or they have a very broad field. So we think that this is going to be and we're putting quite a bit of investment into this. We're trying to see what's happening to these cells in different mouse models on the one hand, and we're trying to also come up with spatial tests for testing humans. So that's the end of my talk. Let me briefly summarize. The hippocampal formation, we believe, provides something like a cognitive map, which can be used to identify the animal, the person's current location, and to navigate from one place to another using any available route. And it's a very flexible system, and that's part of the heart of it. We now know quite a bit about, and I haven't told you all of the different cell types, I didn't have time, but we know a lot about the different cell types which go to make up the map. We also know, for example, that there are cells which represent objects in particular locations as well. It's no good having a map if you don't have any information about what you're going to find in different places. And we think we can use this understanding to both devise spatial tests for humans who are at risk of Alzheimer's and to identify them at a fairly early stage, and also to use it to create mouse models and to test them to see what's actually going wrong in the biology, exactly what's happening when these toxic proteins invade the cells, how they're transmitted from one part of the brain to another. And finally, I'd like to thank not only my collaborators, that I've mentioned many of them, but also our funders. We've been very, very fortunate over the years to get extensive funding from the Wellcome Foundation and the Gatsby Charitable Trust. And they've combined to provide us with enough resources to build a building which is shown in the lower right-hand side, the Sainsbury Wellcome Centre, whose remit is to actually study the circuitry in different parts, not only in the hippocampus, but the way in which the different cells interact with each other, and to see if we can understand how that works at a network level. And finally, I've been at UCL my entire career since I came here many, many years ago, and I'm very grateful for their having housed me and, dare I say, put up with me for these years. Thank you very much for your attention.